So I'm going to go ahead and start. I'm very pleased that we have Dr. Ray Suhandynata with us. He's assistant professor at UCSD in the Department of Pathology and the Skaggs School of Pharmacy and Pharmaceutical Sciences. He serves as the associate director of the CMCR Reference Lab, which conducts methods development and testing in collaboration with the Department of Cannabis Control. He also provides clinical oversight for the clinical chemistry and clinical toxicology labs at UC San Diego Health. So part of Ray's research is focused on development of applications of mass spectrometry in forensic, preclinical, and clinical labs, ultimately leading to an interest in evaluating the utility of forensic toxicology to help increase the predictive values of officer-performed field sobriety testing. So Dr. Suhandynata, the work he and Dr. Fitzgerald have done has been really integral to our public safety and medicinal cannabis research here at CMCR. And I'm very pleased that Ray has offered to present to us today. Ray, it is all yours. Thank you.
Yeah, thanks. Thanks for the really nice introduction, Tom. Let me see if I can share. I share it right now, right? I can share.
Yeah, yeah, yeah.
Let me see if this works. And then are you seeing presenter view or do I need to swap?
Okay, you need to swap.
Yeah, 1 second. Good?
Nothing swapped.
Nothing swapped yet? Oh, really? Oh, that's weird. How about now?
And now we just have you.
Oh, great. Technical difficulties. We did this before we started. Okay, let's try again. Which, good?
Perfect. We're good.
Okay. Okay. Well, yeah. Thanks again, Tom, for a nice introduction, and thanks everyone for joining. So today I'm here to talk about the title of the talk, Still Testing Positive Implications Persistence Led THC Levels in Regular Cannabis Users. To start us off, kind of what kind of drove the synthesis of this sort of particular evaluation of a large study that Doctors Marcotte and Fitzgerald and the rest of the collaborators within UCSD kind of started was to look at, increase in national cannabis use and what does that really mean. So really the legalization, obviously, both medical and recreational, has certainly driven up the use of cannabis nationally. I found a pretty interesting study, just kind of looking through the literature, 2024, published by Calkins, looking at that cannabis actually surpass alcohol on a daily or near, when looking at daily or near daily basis among individuals 12 years of age and older in the US. So I thought that was really interesting. Certainly underlines sort of the growing sort of emphasis and importance of evaluating cannabis use, and especially in the context of DUID or driving under use influence of cannabis. So anyways, one of the things from this study that was, I'm going to try to get my laser pointer. This 15-fold increase was really kind of key. It's just a huge increase, and it's just the prevalence of cannabis use in the general population is very significant. So one of the things also that I think is important to talk about, cannabis legality by state. Basically, it's a mess. So adult use, recreational use 24, including the District of Columbia, as well as medical use is approved in 38 along with the District of Columbia. And you can kind of see there's different colors here, legalized, medical, and decriminalized, which really means that it's medically legal, and but it's decriminalized as in they won't enforce it, basically is my understanding. I'm not a lawyer. Medical, decriminalized, and then there's also fully illegal states here. But in the context of DUID or DUIC, it's really important to kind of look at this this map, which is really looking at state marijuana impaired driving laws. There's this thing with zero tolerance per se laws, which really means you're per se impaired if you have a certain concentration of cannabis, THC, in blood, usually. And if you take a look at this map, 12 states have zero tolerance for THC completely, which means, and I'll kind of emphasize this more, as technology improves, this becomes more and more ridiculous. 5 states have specific per se limits for THC, and Colorado has a permissible inference law for THC, which is, I won't get into that because I'm not a lawyer, but it's kind of a unique thing that Colorado does there. But if you kind of take a look at this and kind of look kind of backwards at the last map, there's some things that really don't make sense. And you'll see that these states are the ones that they're legalized. These are legalized for recreational medical. These are the ones in red asterisks, and then medical approved in these states. The interesting thing there is that these are zero tolerance states. And what that means is that if you are found driving with any concentration of THC, that's not okay. Basically, you are in violation of the law. And as technology improves, as our detection limits for THC and any other drugs of abuse improve, it becomes more and more of a ridiculous thing. Basically, if we find any THC, no matter how small, you're driving under influence. So that's just a really important key thing here. So one of the things that I'd like to highlight is THC concentrations persist in blood and plasma. That's something that's pretty well known. Others have studied this. There was a study by Desrosiers, which really by Marilyn Hustis, and looking at cannabis use, heavy cannabis users in a secured research unit. So just to quickly summarize, very heavy users of cannabis, up to some individuals in this study had 21 joints per day, just not that representative of a normal population of normal use. I mean, I'm sure we may know some people that use cannabis this much, especially in the state of California where it's legal, but it's just not a good representation of the average population. There was a report in this study that the highest baseline concentration of THC, that was at least before the study began, they measured baseline THCs in blood, and it was 8 nanograms per mil. And then there's another study as well, Bergamaschi, that looked at, again, THC and its metabolites in 30 heavy cannabis users. And I just kind of, the theme is that these are really heavy users, 10.2 joints smoked per day on average for this study. Just something to keep in mind. The previous work really was done just because of the nature of it's a scheduled drug. These were heavy users. So as you know, Dr. McGaugh has talked about, and Dr. Fitzgerald's talked about, there's a lot of interdisciplinary collaboration here at UCSD. I've been fortunate to be involved with it, and looking at what is the largest randomized clinical trial of cannabis use and its effect on driving has been really cool, and also just working with others and CMCR to look at the pharmacodynamics has kind of given me a unique perspective. But the conclusions, or not the conclusions necessarily, the evaluation of baseline THC concentrations is really derived from this study. So the study really was supported by something called Assembly Bill 266, and it involved looking at 191 regular cannabis users, and that's different than heavy cannabis users. And it was a parallel design, blinded, double blinded, and really, there's this thing called smoke ad lib or ad libitum, and basically smoke as you would to get high. And that's kind of the instructions to our participants. They would smoke as they would normally, and rather than being forced to take a dose. And there's 3 different doses of THC that were given that are shown here, and it was pretty well distributed among placebo, 5.9% THC and 13.4% THC. They were assessed throughout the day, which I think is important. One of the things that I didn't really talk about before, but these are observed, so we kind of know that they're not also using, off in the bathroom or anything like that. There's some control there, and I think that's really important to kind of derive some of the conclusions that we got. But importantly, because it was a driving study, there was driving simulations, which I'll kind of briefly cover, as well as different matrices that we also took a look at, as well as having officers perform field sobriety tests. Really, the purpose of the study was identifying sort of impaired driver. Well, not the purpose, but one of the things that we did was then try to identify impaired drivers. It was a real world determination in the sense that we use real officers for this, which is really cool. And these are specifically drug recognition experts. So a special class of special training of officers. And in combination with the simulator, which was essentially how we kind of assess driving, as well as interviewing the participants. As I mentioned, officer performed field sobriety tests, as well as really kind of my kind of wheelhouse, which is more toxicology and analytical chemistry. This is really cool. I think that other studies have done these comprehensive, well, not necessarily comprehensive, these field sobriety tests performed in a way that a drug recognition expert would do it, but we use real drug recognition experts, which was, I think, really a key kind of thing that we got from this study. Not to belabor too much, but here's the inclusion criteria. Dr. McCraw has talked a lot about this, so I'm not going to spend too much time, but just so you know, we kind of focused a little bit on more regular population. We tried to take a look at individuals that have been driving a reasonable amount, so you basically take away some of the maybe bias of, say, a new driver or someone that hasn't driven for a long time. We did also take a look at oral fluid THC concentrations on the testing day to kind of rule out recent use, as well as we did do a urine drug screen, which is pretty key there, I think, just to make sure there's not any confounding drugs that the participants were using. This is, you've probably seen this before, but this is just the idea is that we are randomizing them. We give them 700 milligrams of generally the placebo, 5.9 or 13.4% THC, and the participants are allowed to kind of essentially smoke as they would. Very cool. I think this was a really key thing for the study, was to do the driving simulation. There's this sort of like iPad that they're using to kind of look at distractors, and that's not really my wheelhouse. Dr. McCaw can tell you a lot more about that, but I thought that was really cool. This is kind of how driving was assessed. So kind of to bring it all back, kind of what we're going to talk about is this is the whole outline of the study, but we're really going to be looking at baseline THC levels, how they kind of interface with per se laws, zero tolerance laws, and I mean, what kind of data and conclusions that we can derive from our study that say, do they make sense or do they not make a lot of sense? So some little more background is that, and this is more kind of what I'm very interested in, is analytical methods. That's what I kind of was trained to do. We do all our measurements by isotope dilution, liquid chromatography, tandem mass spectrometry. And this is how forensic labs do it, especially with chain of custody, and it's considered defensible. Which was important I think, to do the study in a rigorous way, so that way you could take some variables out. And I think an analytical variable is something that I'm not really a fan with. So validating your assays are important. So we use triple quadrupole mass specs, which is essentially what many forensic labs will be using. And for those of you that have never seen a liquid chromatograph, I'm not going to spend too much time talking about it, but these are at our limits of quantitation. So we're measuring these analytes that are being analyzed by tandem mass spectrometry, and this is the signal and noise that we're getting. So the idea is that we have a good idea. We're not measuring noise, and that's really the takeaway here. So if you really like this stuff, we continue, and I think we'll continue to publish our methods. We have a good standing fellowship. Dr. Fischel and I run a clinical chemistry fellowship, and I think a lot of fellows have benefited from sort of this interdisciplinary collaboration with CMCR, and so we'll continue to do that. So bringing it back, per se, driving under the influence of cannabis statutes. So let's talk a little bit about that. So it's prima facie evidence of impairment, meaning that if you exceed a certain concentration in whatever body fluid that you're measuring, then and that if there's a per se law for that, then you are per se impaired. So there's some confusion here. Some states use 2 or 5 nanograms per ml of THC in the whole blood. Some states have zero tolerance, which I think is, I'll continue to talk about this, but the zero tolerance thing is something that really bothers me personally as an analytical chemist. Basically, it means that I could find, as you develop methods and you get more and more sensitive, you find anything. Maybe you get to the point where you're finding, instead of nanograms, you're finding picograms, single-digit picograms per mil, and you could make an argument if you just read the law that that's you're in violation. I think that's kind of getting ridiculous. So inactive metabolites are also looked at, which there's reasons to not do this from a legal perspective, but also I think historically it was used because it was easier to detect inactive metabolites. So that's kind of one of the reasons why that's done. And really the question that I have for everybody to think about is what data supports the use of these cut points and what's out there right now. We do have some data that's looked at delta-9 THC pharmacokinetics and pharmacodynamic effects. I'm not going to spend, this is not a pharmacokinetics and dynamics lecture. So the idea here that you can see is that tissue distribution plays a prominent role. Essentially what you're following here is there's these on the y-axis is THC concentration, on the x-axis is essentially distribution of THC in the body in time, in hours. And what will happen is, as a dose of THC is consumed, as someone consumes THC, distribution in the blood compartment, then going into the brain compartment, then to high perfusion tissues, low perfusion tissues, and fat. And this is a complicated interchange of, it's a multi-compartment PK, and we have Dr. Momper and Dr. Caparelli who's here. They can tell you a lot more about this than I can. But one of the things that comes out of this is there's something that's, I just want to point out in the context of this talk, is that inhaled bioavailability, which is the amount of drug or analyte compound that's available that's bioavailable, kind of increases, at least there's correlation with baseline THC concentrations. And so there's some inference there with if a regular user is using THC, then they'll have higher baseline concentrations. There's data to show that. And really what my takeaway from that is that more frequent users of THC are better at smoking it. And I think I don't think there's more than a correlation, but I think that's kind of what my takeaway. The pharmacodynamic effects, there's a cognitive and psychoactive effect, euphoric, enhancement, sensory perception, decreased learning, attention, process speed, psychomotor abilities, which is kind of that's the reason why there's concerns with driving. That's really the conclusion there, and increase in heart rate and blood pressure. So that's sort of where this whole driving, why are we concerned about individuals using THC and also driving? That's kind of trying to tie it back together. And the last thing I'll kind of say, it has a very long terminal half-life. Specifically because of the way that the drug gets distributed. So baseline concentrations, it's not that surprising that you have these long elimination of this drug. So, all right. So one of the findings that Dr. Kaufman has published on this is that, and I thought was really interesting, was that there's zero correlation between how well individuals drive in the study that was done with regards to concentration of THC. What you have here on the y-axis is a composite driving score. The higher, meaning their individuals are doing worse, and the lower, they're doing better. And on the x-axis is concentration of THC. You would expect if there's any correlation, you get some correlation. This is really just a shotgun scatter. This is looking at blood THC concentration 30 minutes post-smoking in the THC positive group only. The takeaway there is that these results don't at least partially support that, indicate that per se impairment is not really something that we should think about with regards to this drug, as opposed to something like alcohol, which you can make arguments maybe that, but it's eliminated a lot more predictably. So let's, again, baseline THC. This is from a study back in 2021 from our group, looking at baseline THCs in our study. These are whole blood concentrations. The concentration is plotted in nanograms per mil on the x-axis, and there in the study we had differentiation between frequent users and occasional users. And here you have THC, 11-hydroxy THC, which is an active metabolite, as well as an active metabolite, carboxy THC, and glucuronidated carboxy THC. Really, if you look at the distributions, you kind of see in a frequent user, you kind of have higher baseline concentrations, and then an occasional user, that's not surprising. But if you kind of look at this, it's a little concerning here, right? So what of this data that you can see here supports per se for driving under the influence of cannabis? And there's this individual out here that's at around 16 nanograms per mil that really bothered us when we looked at this data set. It was something that always stood out. It's the same individual here. This individual for sure would be in violation of all the per se laws because they're clearly an outlier, and we wanted to take a look, is this something that's what happened here, right? So here's a histogram. So this is all of the AB266 study. We plotted the baseline THC concentrations as the frequency of the count, and then baseline THC concentrations in whole blood, and this is the distribution, and there's that one individual here at 16 nanograms per mil. Clearly an outlier. So we are here to talk about this individual today. So first question that we had was, did this individual smoke, or did all of our individuals smoke the morning of the study? So we kind of took a look at this in a way that looked at the final THC concentration, which is around 5 hours after smoking, to baseline THC concentrations. And what you see is that the mode is 0. So this is very indicative that, for sure all these individuals came back to baseline because the difference is 0. If you went back to your baseline, you should have no difference, right? And then other individuals, now there is analytical bias in the measurement, there's an imprecision in the measurement, but this is the active drug group, and I do think that many of them are on their way to returning to baseline, and there's some that may be on their way. But the difference in THC, this is one nanogram per mil. The higher the initial measurement, we typically think our assays are about plus or minus 20%. That's how we validate our assays. So there is some it's not like a hard and absolute rule. So it's just 20% of within the two measurements. So we basically felt this data kind of showed that most everybody comes back down to baseline because the difference is reasonably close to within 20% of the original baseline measurement of a participant. So median difference in this was half a nanogram per mil. We felt pretty good about that. So at least at a macro level, we were feeling like and interpreting that our population came back down to baseline. So baseline THC concentration in blood for this individual. What about, oh, weird, I just put it backwards, okay. Placebo THC doesn't change. This is we also looked at the placebo group here. This is the final THC concentration minus the baseline THC concentration. And again, also no change. In this case, this kind of shows the noise, right? You can kind of see in these individuals, they really didn't get active drug. There's still that scatter, but there is some less of this there's not this hump that was shown in the active drug group. So the placebo really is the control. You see that this kind of looks, this would be like truth, right? So there is a little bit of the population that was still coming back, but more or less they came back to baseline. Still, we still felt like there supports no morning of use in our population. Otherwise, we would see a hump. And longitudinal plots of THC, this is just contextualize everything. This is also another study we published in Journal of Animal Toxicology. This is just the overall PK. Frequent users and infrequent users generally look the same in terms of their the kinetics here. And there is a peak shortly after smoking, which you would expect. It's a dose response. It's what we're taught. So the takeaway here, though, which we talked about, is that even though we gave essentially more than double the dose in some individuals, which is not what we're taught. You should, if you give double, you should get double in terms of a whole blood concentration, which is on the y-axis. It kind of highlights this concept of self-titration. We asked our participants to smoke as they would as they got at home, which is ad libitum smoking, and this kind of reinforces these things that were published by Marilyn Hustis, which is the self-titration is a thing. The participant or a user of cannabis smokes cannabis to the point where they get high, and they are good at that point. So it's an interesting concept, and it's not really what we're taught, but classically. But it is. I'm beginning to get more behind it, anyways. So highest THC returns to baseline. This is back to our individual. This is kind of the star of today's talk. Here we have on the y-axis, THC concentration in whole blood. This is this individual's kinetics through time in this study. So we kind of extracted this individual and kind of plotted their THC concentrations in whole blood over time with the study. And you can kind of see it looks the same, looks like everybody else, which is part of what we were trying to convince ourselves is this person's baseline concentration really just 16.2. And you can kind of see it goes up, comes down, it gets cleared, and it really does look like this individual, she comes back down to baseline. So again, not over days, this is really only after 5 hours. So you can kind of see that their baseline concentration is, at least, even though it's an N of 1 for this individual we're looking at, it does come back down to baseline. Interesting things about this participants, they drove better than average at baseline. Their composite driving score, in the context of just the whole study, they did really well overall. And I guess just to kind of reinforce the composite driving score, what it means. So better, worse, and this is over time. The real key thing then, that I got from this particular chart is that to look at the placebo. The placebo, if there's any kind of learning effects, then it would kind of go up. But what you really see, and I think that was a really smart thing to how Dr. Mercado really designed this, is really to kind of convince there's no learning effects. Showing that over time, the composite driving score, if there's learning effects, then you'll actually see the placebo, which is the control, get better and better. There would go this way. But there's a directionality. So in this case, it's flat. So elevated THC during driving, baseline THC during driving, that's something that we're trying to hammer home here. The baseline composite driving score, which is again on the y-axis, we kind of looked at this in the context of our analytical method, but we also kind of wanted to look at per se cutoffs in whole blood. And first we asked, well, is there any difference from individuals that we could quantify the THC levels in whole blood versus individuals that we couldn't quantify the THC in whole blood? And that just basically means like this individual, in terms of a zero tolerance state, positive for THC, negative for THC, and the answer is there's no difference. What about in a 2 nanogram per mil THC state? Again, no difference. And what about a 5 nanogram per mil THC state? And the answer is no difference. So these would be per se cutoffs. And really the takeaway for me looking at this data was that there is no support for THC per se or zero tolerance cutoffs and simulated. I mean, this was simulated driving, so that is a limitation. This is not on-road driving, but at least in the study, we did not see any difference, and I think that's an important thing. I think that's an important finding, and again, because you have individuals that could have used THC several days ago or even weeks ago, if that's just their baseline, that would essentially be incriminating. I am going to try to pull this back more to the field sobriety tests that were done. I think that is an important thing to at least contextualize with this. How did the majority of the participants in this study do on the field sobriety test? And what we have here is a table showing the THC group and the placebo group, and really the first exam performed by the drug recognition experts and the field sobriety tests that were done, and then how they classified each of the participants. And as you can see, on the THC group, they saw that 81% of the THC group they classified as impaired on the first exam, and they said 19% wasn't. But really, one of the interesting things from this study was that the placebo group, they called half of the placebo, which didn't get active drug, remember, they called them impaired. So you could look at it as a really good placebo. I think there's considerations there, and I think forensically that's not okay. Like for us as a forensic, from a forensic toxicology point of view, we want, that's kind of where we believe that toxicology has a real role. So this is one of the key things that I always look at with this study. So what is sensitivity, right? And in the context of tests out, like test classification, and we think about that all the time for patient tests and things like that, but also in the context of forensics, how well does a test classify truth and true positives and true negatives? And in the case of the THC group, we considered those true positives. They used drug. The officers, as I mentioned, performing the test got 80% of those right out of 100. It's not 100, but we basically got 81 of them right, 81 out of 100 because it's a percentage. And if we apply toxicology cutoffs in whole blood, you'll actually see that this goes down, and that makes sense. So you're applying a second filter to the 81%, and basically if you require to have half a nanogram per mil of THC in whole blood, 2 nanograms per mil of THC in whole blood, or 5 nanograms per mil of THC in whole blood, you'll get worse and worse detections. And basically, that's what you would expect. But let's take a look at specificity, which is basically a test's ability to classify a true negative. So if an individual really didn't use THC, let's see how our test does in classifying that individual. And as I mentioned earlier, 50%, they got 50% of these individuals as impaired when they didn't really use THC. So we consider that as a false positive. And if you start applying whole blood THC concentration cutoffs at half a nanogram, 2 nanograms, and 5 nanograms, you get better predictive values, or you get better specificity because you're applying another cutoff that makes it more specific to whether or not these individuals use THC. And we sort of what we kind of focused on in that publication was the 2 nanogram per ml cutoff, which is a per se cutoff. And you can kind of see whole blood works not the best. 60-ish% sensitivity, meaning you're missing 40%. Well, not really 40% because it's really it's a portion of the 80%. But you're still calling about 13% of the individuals that did not use active drug as per se impaired, which from my perspective, that's too much. So that was one thing we looked at, and I think that made us think, well, we also collected oral fluid, we also collected breath, which didn't work as well, but oral fluid was sort of kind of what we came away with as potentially something to really kind of focus on. So here I'm kind of switching gears a little bit to show you kind of how oral fluid performed using the same types of metrics. Oral fluid THC sensitivity, again, 80%. This is what, remember, the officers are saying these are of the THC group, that's what they think is impaired. So they got 81% right. And if you start applying these same cutoffs, you're kind of looking and seeing when they drop off. And there's a big drop off between 2 and 5, almost no drop off at, I think there was no drop off at half a nanogram per mil, and then a little drop off at 2. And but really the important thing was, when does the specificity, as in when do we start eliminating our false positives? What happens? When does that happen? And in this particular cohort, we were looking at a 2 nanogram per mil oral fluid cutoff for THC. And that's what we want to see, because then, now, no test is perfect. This is when we do, if you test enough, you'll always get a false positive. No test is perfect, but at least this looks a lot better than the 13 point something percent that we had calling incorrectly as using THC, or impaired, I should say, because they had not used THC at all. So that was a highlight. 0% placebo patients. That's kind of participants in our study. That's kind of what we were going for. And so the conclusion was, at the 90-minute post-smoking time point, we could say that we sort of eliminated. Well, we in fact we did, at least in our study, are eliminating false positives. So I'm going to wrap up a little bit with study limitations. There was no absolute confirmation of period of abstinence with self-report. We did not sequester aortic persistence in a closed facility prior to the study. We did not. That just wasn't part of the study. So there's some limitation there. But we did remember do a Draeger, which is oral. We took a look at oral fluid THC, and that they met that criteria. Not real world driving, in the sense of not actually in a moving vehicle on the road. Simulator performance. That's how driving was assessed. So a little limitation there. But we do have some conclusions that I think, even in spite of those limitations, that we can kind of come to. So there we had many regular users of cannabis exceed the zero tolerance and per se THC cutoffs days after last use. 40% exceeded those zero tolerance cutoffs, which is a lot. And I think I've sort of tried to re-emphasize why I don't think that's great. As technology gets better, that number within just the same study that we're doing will go up. It'll just keep on going up, because you'll just be able to detect lower and lower and quantify lower and lower amounts of THC. Nearly 25% exceeded that 2 nanogram per mil, and approximately 5 exceeded the 5 nanogram per mil, which, I mean, that's tough. You're basically saying that if you're a user of cannabis and you just happen to go into a per se state and you haven't used cannabis for a while, you drive, that's kind of what the implication is. You're sort of putting yourself at risk. Baseline concentrations reflect that sort of steady state, as it has mentioned, showing the PK distribution curves of in terms of how it distributes in the tissues. There is an exchange between the compartments, and that kind of that steady state of the drug is sort of reflected in there. Baseline THC concentrations are higher in frequent versus occasional yields. Again, it makes sense, but that is something that I think we observed, as well as many others have as well. And I think one of the things that is important from our study is these are more representative, I think, of a normal population of recreational use. Driving performance not different. I think that's a good, really important finding. Regardless of different cutoffs that you're using, we didn't see any statistical difference in driving performance. And really, kind of the things is all the data that we have so far, nothing that, in my opinion, I've seen supports using per se DUID cutoffs. But a little bias here because my background is in analytical chemistry and toxicology. But I think using toxicology helps get the right answer. If the goal is to limit false positives, classification accuracy, that's what you're going for. If you're trying to say THC is the reason, if the field sobriety test and the drug recognition officer saying this individual is impaired by cannabis, and the toxicology saying they don't have any cannabis, then I think that's important, and I think this should be included. So, wrapping up, future directions. I think we still have ongoing studies. Dr. McCott's sort of led the charge doing this and looking at different effects of cannabis in the context of a polysubstance abuse, alcohol, and there's some hope that we get some studies going there. And there's a really exciting study that we're kind of launching. I think it's already started, that we have essentially looking at the effect of real world cannabis products in dabs, vapes, concentrates, and really, I think a huge shout out there to the CMCR team. Dr. Cheryl and I went down to HNRP maybe a month or two ago, looking at the van they have. It's really cool. Having the simulator in there, I think it took a lot of engineering. So that's a really exciting study that's sort of on the horizon. So with that, wrapping up, I'm just part of a team here, and Dr. Cheryl and Dr. McCott really kind of put this together. It's a lot of people have been involved. Everyone on this list has done remarkable things, and with that, we've had extra consultation from Marilyn Hustis, who's been a leader in this field, and as well as a lot of funding from the state, and also obviously support from CMCR and the rest of the UCSD. So with that, I think that's it.